Chapter 8, the Special Senses. In this chapter, we're going to be primarily concerned with the special sensory receptors that I touched on in the last chapter, and we're going to talk about the organs which house these receptors. Here are the primary objectives for this chapter. If you want to read them, just pause the video. Section 8.1, the special senses tell us about our environment. So we have two types of basic senses. First, there are the somatic senses, which involve the general sensory receptors and the visceral receptors that we talked about in the last chapter. And different kinds of these receptors can be found in more than one place in the body, or on more than one place in the body. And an example of such receptors would be those which help us to coordinate muscle movement and maintain body temperature, and also those that allow us to sense that we're being touched, and then there's several others. Then there's the special senses, which are extremely sensitive and involve receptors that supply us with detailed information. And there's three basic types of these. There are photoreceptors, which sense light, and we use these for vision. There are mechanoreceptors in the ear, which we use for hearing and balance. And then we have chemoreceptors for smell or olfaction, and taste or gustation. And these are obviously in the nose and on the tongue. And then sensory adaptation is the process by which you can become accustomed to or desensitized to an unchanging smell, sight, or taste. The perception of that sense simply decreases to the point where we are not aware of it any longer. So I don't know if you've seen those commercials for the air freshener Glade, but sensory adaptation is what happens when they say you go nose blind and can't smell a scent that's obviously there to others. The word olfaction refers to our sense of smell, and this occurs in the upper chambers of the nasal passages on the roof of the nasal cavity. The olfactory cells extend from the olfactory bulb, which, if you'll recall from the last chapter, is at the end of cranial nerve 1, through the cribriform plate and into the mucous lining of the nasal cavity. Receptors are actually small yellow patches of olfactory epithelium in the lining of the nasal cavity. And each of these receptor cells is a modified neuron that ends in approximately 6 to 12 olfactory cilia, and each of these bear at least one of many thousands of very specific olfactory receptors. So when a receptor binds a specific odor molecule, a sensory impulse is created, which is then sent to the olfactory bulb, and then it's sent to the brain so that we can interpret what we're smelling. Here's a picture of the anatomy of our nasal passages. So, olfaction isn't really all that complicated of a process, and it's kind of hard to tell in this diagram, But down at the bottom of part B, you can see the odor molecules which bind to the olfactory receptor neurons, which are the ones in blue, and then the neural impulse is sent up the axon of these cells to the olfactory bulb neurons in the olfactory bulb, and then the impulses are sent to the brain. And I have a better picture of this in the next slide. Here's another diagram of olfactory anatomy, which isn't in your book. And this just kind of gives us a better look at how the odorants or odor molecules bind to the receptors. And you can see these odorant receptors there in the picture at the bottom of this diagram. And there you can just kind of see how the molecules bind. And it's pretty much the same sort of picture. It's just a little bit easier to see the actual receptor sites. And don't bother memorizing names off of this slide. I'd rather you take the names from the last slide just because they're more simple than this. But anyway, moving on. Gustation or gustation refers to our sense of taste, and it's very similar to olfaction as they both involve chemoreceptors. So the taste receptors are found in roughly 10,000 taste buds, which are on the surface of our tongue, and these are located in small bumps called papillae. And there are actually 5 kinds of taste receptors, each of which responds to a different class of compound. So there are sweet taste receptors, which respond to glucose and other sugars. We have taste receptors which respond to salt or sodium chloride and possibly other salts. We have receptors which respond to acids or substances with a high hydrogen concentration, and this is what gives us a sour taste. We have taste receptors for sensing hydroxide in bases or more basic food or foods with a high pH, and these foods taste bitter to us. And then finally there are umami receptors, which are called that because they were discovered by a Japanese man, so naturally he decided to put his name on it. And this is more of a savory or meaty flavor. And these receptors sense glutamate, which, if you'll recall, is an amino acid. So one type of glutamate these receptors would sense would be monosodium glutamate, or MSG, which is pretty much just a glutamate salt, really. The taste buds collectively respond to only 5 classes of compounds, like I said, and each individual taste bud only responds to one type of compound. When the taste bud receptor cells are stimulated, they send this information to the brain, where the overall taste of food is determined. And many differences in the taste of food is actually due to the involvement of olfaction. And a lot of that's because the nasal cavity is actually connected to the oral cavity, and the uvula, or that flap of skin that hangs down in the back of your throat, actually acts as the door between the two passages, which gives some of the molecules or odorants in foods a way to drift up into the nose, and that's how olfaction is involved. Here's a picture showing your taste bud and tongue anatomy, and it's just a good diagram to look at to tell the difference between taste buds and papillae. So over here in this tongue cutout, you can see where it points to 3 different kinds of papillae, and you don't need to know the 3 different kinds. Just know that they're bumps on the tongue which contain taste buds. And over here in part C, you can kind of see more of the taste bud anatomy, and these have pores in them and a few other things, obviously, and then a sensory neuron. And you don't need to know any specifics of this, I don't think. Just know the difference between papillae and taste buds. And over here in part A, it shows the taste zones for 4 of the 5 types of taste receptors, and this is just where they're primarily concentrated. And apparently there aren't yet any diagrams of where the umami receptors are just yet. It's still a pretty new discovery. Moving on to our sense of hearing, which involves membranes, bones, waves, and hairs. And obviously the organ of hearing is the ear, which has three basic functional parts. First, there's the outer ear, which includes the pinnae and the external auditory canal. And yes, I don't know if I necessarily spelled that right, but the word is pronounced pinnae. And these structures funnel sound waves into the ear. There's the middle ear, which includes several structures such as the tympanic membrane or eardrum, ossicles, and ends before the oval window. That is to say that this part of the ear becomes the inner ear after the oval window. The ossicles are actually tiny bones in the ear, and these are the malleus or hammer, incus or anvil, and stapes or stirrup. When the tympanic membrane vibrates, this causes the ossicles to move, which vibrates the oval window, and these ossicles are joined together with what are called synovial joints. And then the inner ear includes the oval round window, semicircular canal, vestibular, cochlear, and auditory tube, and we'll talk about this in more detail in the following slides. Here's a diagram of the anatomy of the ear, and we'll hit on these a little bit more in a second. But anyway, the eardrum is that membrane shown in blue, and then there's the malleus, incus, and stapes, and as those are their proper names, those are the names you're going to be using to refer to them. And then there's the stapes, which is connected to the oval window, or rather it touches the oval window. And then there's the round window and the semicircular canal, the vestibule, the auditory tube, which is actually a tube that connects your ear to your mouth, more or less. And then there's the cochlea, which is a very important organ in hearing. Now we're going to talk more about how you hear sound. The entire middle ear and inner ear are within a hollow portion of the temporal bone. So the mechanical waves at the tympanic membrane are transferred to fluid waves at the oval window. And now here's more of a definition of some of the parts that we just looked at. The auditory tube is what connects the middle ear to the nasopharynx, and this ensures that the air pressure on either side of the tympanic membrane remains the same because we've got air pressing on it on either side. And then the cochlea is actually a coiled tube, and it looks very much like a snail, I guess. And this is filled with fluid and is located in the middle ear. And the cochlea has three major parts. There's the vestibular canal, which is the uppermost compartment and is continuous with the oval window. There's the tympanic canal, which is the compartment that rounds the bottom end of the cochlea and ends at the round window. Both this canal and the vestibular canal form a U-shape, fluid-filled passage for the pressure waves. And then there's also the organ of Corti, which converts mechanical vibration into sensory input. The tectorial membrane lies on top, and this is a flattened membrane resting over the hair cells, which have stereocilia. And these hair cells are directly linked to the vestibular cochlear nerve or cranial nerve 7. So after the sound wave deforms the tectorial membrane, this bends the hair cells, and this bending of these hair cells creates a nerve impulse in that particular hair cell, and then this nerve impulse is sent to the brain. And as with any other special sense, the impulse is then interpreted by the brain as a particular pitch. And each part of the tectorial membrane is sensitive to a different pitch. Lower frequencies vibrate the organ of chording near the tip of the cochlea, and higher frequency sounds cause vibrations at the base. And obviously, hearing involves vibration of most all of the parts of the ear, including the ossicles, which I should have probably mentioned earlier. Not only is the ear an organ of hearing, it is also an organ which allows us to balance and sense gravity, and also to know if we're in motion or not. So static equilibrium is the physical response to gravity, which tells us which direction is up or down. And the two structures used for this are the utricle and saccule, which are located in the vestibule of the inner ear. And these two structures also have hair cells, just like the cochlea, and the bending of these hair cells initiates the nerve impulse. They contain two gelatinous blobs situated at right angles to each other in the vestibule, and these blobs are called the maculae, and they are held in place in these structures by hair cells. Each maculae contains tiny pieces of bone, which are called otoliths, that respond to gravity. The utricle and saccule are also arranged at right angles to each other, so that when the head is upright, one of them is vertical and the other is horizontal. So the hair cells bending causes the nerve impulse to be generated, and this impulse goes to the part of the brain which is responsible for static equilibrium. As our head position changes with respect to gravity, these impulses change in frequency and direction. So here are some diagrams of the structures responsible for static equilibrium and dynamic equilibrium, which we're going to talk about in a second. And you don't really need to know a whole lot about these, just know more or less what the utricle and saccule are for, and it'd probably be helpful to remember that these contained hair cells and otoliths. And then over here at the far right, we have the structures of dynamic equilibrium, which involves the semicircular duct and the ampulla, and this is made up of the cupula, hair bundle, crista, and it's connected to the ampullary nerve, which we're going to talk about in the next slide. And same thing with the last one, just know what the primary structures are for. And I'd like to point out that this picture says that the vestibular branches, or rather the vestibulocochlear nerve, is nerve 8. So I'd just like to correct some of the slides without audio. It should say nerve 8, not nerve whatever it was. I think I had 2 I's before V, and that's not even a number. So dynamic equilibrium is a response to changes in motion. And this is actually what allows us to detect acceleration or deceleration of our head. The sense originates in 3 semicircular canals, one for each plane X, Y, and Z. The fluid in each tube in these canals rocks in response to acceleration in its particular plane. So the ampullae is a swelling at the base of each canal, which houses the dynamic equilibrium receptor, which is called a cupula, which is a flame-shaped gel with the hair cells embedded within it. The cupula, or rather the fluid in the semicircular canal, rocks through the swollen base of the canal, and then it pushes on the cupula, which bends its hairs. And this sense is responsible for allowing us to feel ourselves going up or down in an elevator. Section 8.2, vision is our most acute sense. So since we have a whole section devoted to vision in this book, you kind of get a sense of how important it actually is. And it is our most used and most important sense, and makes up a large percentage of our brain so that we can interpret visual images. Our eyes actually work like a very sensitive camera, and they regulate the amount of light that enters the photoreceptive area and then capture that light as an image which is sent to our brain, and is actually sent to the occipital lobe of the brain as a nerve impulse, and then the brain interprets the impulse. The organ which houses the photoreceptors which sense light is the eye, and it has 3 layers. First, there's the sclera, which is a fibrous layer which includes both the white of the eye and the transparent cornea, which is over the lens. And the sclera is bathed in tears from the lacrimal gland, or you can call them the tear ducts, I suppose. The sclera provides an attachment for the 6 optical muscles, which are the lateral rectus muscle, the medial rectus, the superior rectus, and the inferior rectus. And these muscles are responsible for moving the eye up, down, left, or right. And then there's the superior oblique muscle and the inferior oblique muscle, both of which pull the eye obliquely. There's the choroid layer, which is the vascular layer, which houses the blood supply for the eye and contains melanin, which absorbs light. And this melanin is actually in the iris, which is a circular muscle that regulates the amount of light entering the pupil. And people have varying amounts of melanin in the iris, and the different amounts of melanin is what is responsible for the different eye colors. The choroid also includes the ciliary body, which is the structure which holds the lens in place, pulling on it to change the shape of the lens to accommodate for being able to see things near or farther from wherever we're looking. And then there's the aqueous humor, which is a transparent gelatinous fluid which supports the lens and is constantly filtered from the blood canals. These blood canals get constricted in glaucoma, which causes an increase in pressure that can eventually destroy the light-sensitive cells in the retina, eventually leading to blindness. And the third layer of the eye is the retina, which is the layer which contains the nervous tissue and the light-sensitive cells, which are called photoreceptors. Here's a cross-section of the anatomy of the eye. So the sclera includes both the white of the eye and the cornea, which means that it runs the whole outer surface of the eye. Then there's the choroid layer, which is shown in pink, and the retina, which is shown in yellow. And the retina is really mostly at the back of the eye. And you can just use this to study the various parts of the eye. And unfortunately, this picture isn't a very good one of the attachments of the muscles to the eye, so we're going to look at that in the next one. And just note that the optic nerve is located at the back of the eye here, and seeing as where the optic nerve connects to the eye doesn't have any receptors in that area, this is what we call a blind spot. More about that in a minute. So here's a diagram of the muscles of the eye, which is not in your book, and it just shows us a better view of how these muscles are connected to the eye, and just lets us see how they move the eye. So all these rectus muscles allow the eye to move up, down, left, and right, and then the oblique muscles, or specifically the superior oblique, moves the eye downward and outward, and the inferior oblique moves the eye upward and outward. So here are the structures of the eye, and we're going to go over these here just because it's a little bit simpler. So there's the cornea, which admits or lets light in, and also refracts or bends light into the eye. Then the sclera, or the outermost layer, is what provides the shape and protects the inner parts. And here they're pretty much just saying that the sclera is the equivalent of the white of the eye, but we're just going to call the whole layer involving the sclera and the cornea the sclera. And then in the vascular layer, we have the iris, which is a circular muscle, like I said, which regulates the amount of light which enters the eyeball, either by contracting or relaxing to control the amount of light that enters. The ciliary body secretes the aqueous humor, which allows us to alter the shape of the lens to accommodate near or far vision. And then finally there's the choroid, which actually provides the blood supply and absorbs scattered light. So I guess here we've got these layers classified as the fibrous layer, the vascular layer, and the nervous layer. It'd probably help to know them by both names, I suppose. And then in the nervous layer, the retina is the part of the eye which contains the photoreceptors, and these receptors convert light into nerve impulses, which provide output to the brain via ganglion cells, and these ganglion cells form optic nerve 2. And then the lens is what refracts or bends light so it hits the retina. And then we have these two, or rather this one cavity, the anterior cavity, which contains the aqueous humor, which helps to maintain the shape of the eyeball and also supplies oxygen and nutrients to the lens and cornea. And then the vitreous chamber contains the vitreous humor, which helps to maintain the shape of the eyeball and also helps keep the retina flat against the choroid, which is very important, of course. In order to achieve optimal optics, the lens must change shape. Visual acuity is the resolving power of the eye, and this requires the eye to focus the entering light onto the retina at the back of the eyeball if we hope to see anything. The lens and the cornea both focus light rays so that they hit the retina. Now the muscles of the ciliary body contract, which causes the lens to bulge, which allows us to better see nearby objects. In contrast, when the muscles of the ciliary body contract, this causes the lens to flatten, and this allows us to better see objects that are farther away. So just try to remember the difference between what happens when the muscle of the ciliary body contracts versus when it relaxes, because it's kind of opposite of what one might think initially. Accommodation is the actual changing of the lens shape to view objects either nearby or far away. As people age, the lens actually grows more layers, which makes it become thicker and stiffer and more resistant to the pull of the ciliary body muscles. and this results in difficulty focusing on nearby objects. And I'd like to make a correction from the slides that aren't in this video, or that actually I fixed this slide to where it said nearby because it did say far away, and that was wrong. So it's nearby. Typically around the age of 45 or 50, people require reading glasses to treat this farsightedness, which is named this because you can see objects far away pretty well, but you can't really see nearby objects very well at all. Here's a diagram showing how visual accommodation works. So up at the top we're looking at a distant object, and as you can see the lens is flattened, which means that the ciliary bodies are relaxed, and this allows us to focus the image on the retina. And note that when the light hits the retina, the image would be upside down, but after the nerve impulse goes to the brain, we actually integrate the information such that the object appears to be right side up, and that's how we see it, because it is right side up. And then at the bottom we're looking at a close-up object, so you can see that the lens is bulging, which means that the ciliary muscles are contracted, and this allows for the light to hit the retina so we can see that object. Now we're going to talk about 3 common visual impairments, and the first 2 are nearsightedness and farsightedness, and both of these are characterized by the inability of the lens to properly accommodate light. So in nearsightedness, the eyeball has changed shape such that it is too long for the lens to focus the light rays on the retina. And that's not to say that you can't be born with eyes that are the wrong shape, but you tend to develop vision problems. This moves the focal point, or the point where light is focused, to the point where it's in front of the retina, somewhere in the vitreous humor, and this causes faraway objects to appear blurry. In order to correct this, we would use eyeglasses with a concave lens, which would spread the light rays farther apart before they enter the retina, correcting the problem such that the focal point actually hits the retina. And the reason we call this nearsightedness is because you can see things that are close up, but you can't see things that are far away. And this is actually the visual impairment that I have. In farsightedness, the eyeball has changed shape such that it is too short for the lens to focus light rays on the retina. So both of these are problems where the light cannot be focused on the retina. And this moves the focal point behind the retina, causing nearby objects to appear blurry. So we would use the opposite type of lens to correct this, which is a convex lens, which will focus the light rays before they enter the eye, or basically make it to where the light rays are closer together, which is going to move the focal point forward such that it hits the retina. The third type of visual impairment is astigmatism, and with this impairment, the lens is fine, but the cornea is imperfectly shaped, and this results in an uneven pattern of light hitting the retina, which causes some areas of the image to be in focus and some areas to be out of focus. So in order to correct this, we would get a carefully crafted lens that compensates for the uneven flaws of the cornea to correct this problem. We could also get a laser eye surgery just to even out the cornea. Here's a diagram of the common visual impairments. So over here at your left we have a normal eye where the focal point hits the retina as it should. So in nearsightedness, the focal point is actually in front of the retina, such that we have trouble seeing nearby objects which appear blurry. So in order to correct this, we would get a concave lens which bulges inward, I guess you would say. And this allows for the focal point to move back and actually hit the retina. And then farsightedness, the focal point is actually behind the eye, such that far away objects appear blurry. In order to correct this, we get a concave lens, which bulges outward, and this moves up the focal point such that it hits the retina. Then in astigmatism, we have this irregularly shaped cornea, which means that we've got light hitting the retina all over the place. In order to correct this, we get this specially crafted lens to compensate for the uneven pattern of our cornea, which makes it to where we get a focal point which hits the back of the retina. Photoreceptors detect light in the retina. So let's just review these two parts of the eye. So the vitreous humor is a gel-like fluid which fills the large chamber behind the lens and maintains a slight pressure on the retina, which ensures that the retina stays in place and doesn't become detached. And the retina is simply a layer of neurons in the back of the eyeball, which contains these parts. It contains rods, which are photoreceptors, and these function in low levels of light and provide only vague images. These are really just simple photoreceptors that just sense the presence or absence of light. And then the second type of photoreceptor are the cones, and these respond to bright light and provide color vision and the resolution of images. The highest concentration of these photoreceptors is at the center of the retina, called the macula lutea. And within the macula lutea is the fovea, which only contains cones and no rods at all. And then finally we have bipolar cells and ganglionic cells, which are interneurons that carry the action potential generated by the photoreceptors to the optic nerve and eventually the brain. Now, interestingly, the layers of neurons in the human eye actually point backwards because the photoreceptors are against the back of the eye, oriented toward the brain rather than outward towards the source of the light. And funny thing is that the photoreceptors in animals like octopus and squid and possibly other cephalopods point the right direction, and this allows them to eliminate the blind spot, which we obviously have. And we'll talk more about how we accommodate blind spots in a second. And octopi are actually one of my favorite animals just because they're so strange. Now we're going to cover the photoreceptors in more detail, starting with rods. So within these rod photoreceptor cells is a chemical called rhodopsin that is responsible for initiating a neural response or impulse. So when light enters the eye and hits a cone, it eventually hits this molecule of rhodopsin and splits it into its component parts, which are retinol and opsin. And this causes the ion gates of the rod photoreceptor cell, its membrane anyway, to close. This causes ion movement to cease, causes the generation of an action potential, and this signal is eventually sent to the brain. And honestly, your book is very vague on the details of this, and I'm not sure exactly how accurate the couple sentences it has in it are. But just remember that when light hits rhodopsin, it splits into retinol and opsin, and an action potential or nerve impulse is sent to the brain. So just remember that for this little part. Also, rhodopsin is easily bleached, which means that even a slight increase in light can cause it to fall apart and be unable to recombine. And until the light is reduced, rhodopsin cannot regenerate. Here's a picture which shows the anatomy and physiology of the retina. And remember, the retina is the third layer of the eye, which contains the photoreceptor cells. And as you can see, these cells are actually, well, the photoreceptors themselves are pointed towards the choroid, which is really kind of opposite of what one would think would be optimal, such that the path of the light goes through the retina, and the actual direction of visual data processing is the reverse of what one would think it would be. And also you can see sort of how rhodopsin degenerates into retinol and opsin once it's hit with light. So once it's hit with light in this picture, it straightens, it looks like, and the retinol is released from the opsin. And just keep in mind that the opsin is this big purple thing, and that retinol is this little pink thing. And then once retinol is restored and changes its shape back to what it normally is, it binds opsin again. Now we're going to talk about the second type of photoreceptor, the cone receptor. And there are actually 3 types of cones, which are sensitive to different wavelengths of light. And these wavelengths are either red, blue, or green. Cones also contain visual pigments or chemicals similar to retinol and opsin, but obviously with slight variation. Whereas the retinol and opsin, or rather the rhodopsin in rods, tends to fall apart and will not readily regenerate in bright light, the chemicals found in the cones readily regenerate, which allows the cones to respond almost immediately to brightening light. So just for a review, rhodopsin in the rods is bleached when it encounters bright light, and the rhodopsin needs time to regenerate. After the rods resume resume working after regenerating, we can see objects even in near darkness. This is, like I said, cones respond almost immediately to brightening light, and this is the reason you can leave a dark place and still see, but will have trouble seeing after moving from a well-lit area to a dim one. And instead of using the word bleaching for what happens in rods, we can also call it a white flash. Now let's go over the pathway a visual nerve impulse takes when it travels to the brain. So after a nerve impulse is generated by the photoreceptors, after they've come in encounter with light, the signal is sent to the bipolar cells, which then transmit the signal to the ganglionic cells, and the ganglionic cells send this information to the brain via the optic nerve. And like I said before, the optic nerve actually contains no photoreceptor cells. So this is what we call a blind spot, simply because there's no receptor cells there. We can't see any of the light that's might be hitting that spot. So how do we make up for a blind spot? Well, it helps to have 2 eyes. As each of our eyes has a different field of view, the blind spot can be filled in by the information received by the other eye, and then the brain puts this together to give us a full picture. And this is called stereoscopic vision, and this also kind of explains why there aren't really any one-eyed or naturally one-eyed animals walking around, because they would have just this area of space where they couldn't see at all. Vision is so important that it occupies more space in the brain than any other sense. And if you think about it, as our eyes are always open, we're always seeing and we're always having to interpret light information as compared to hearing or smelling or tasting, which we only use occasionally throughout the day. So now let's talk about optic chiasma, which is talking about the physical crossing of the left and right optic nerves. So this means that an image from the right eye is partially projected on the left side of the visual cortex of the cerebellum, and this is the reverse for the left side. And also, when an image or light off an image hits the retina, the actual image is projected upside down and inverted on the retina, such that when the neural information or neural impulse reaches the brain must interpret this information such that it flips the image and inverts it so that it makes sense to us, and so we don't see the world as being upside down. Now we're just going to clarify how photoreceptors generate neural impulses. So first, light enters the eye through the lens, and it hits the retina, which contains the photoreceptor cells, which are rods and cones. This changes the physical form of rhodopsin such that it breaks into opsin and retinol, and this creates the generation of an action potential. The nerve impulse is then sent to the bipolar cells and then to the ganglionic cells, which connect to the optic nerve. The optic nerve then sends the impulses to the brain, where the image can be interpreted. Now we're going to backtrack to hearing, and we're going to talk about how it can diminish with age. And yes, I could have included it in section 1, but I just thought that if you look through the book and maybe read these slides at the same time, it might help to keep them in sequence. But anyway, there are two forms of deafness. There is conductive deafness and nerve deafness. In conductive deafness, sound is simply poorly conducted from the outer ear to the inner ear. Such an example of this would be if the ossicles were prevented from moving easily. So in this case of deafness, we treat it with hearing aids, which can help by increasing the amplitude of the sounds that enter the ear, and would basically solve the problem of conducting the vibrations to the inner ear. Now, nerve deafness, on the other hand, is deafness due to a neurological malfunction rather than an actual physical conductive problem. So in this case, a hearing aid will not help, as the sound is either not detected by malfunctioning cochlear nerves, or the nerve signal is simply not getting to the brain for whatever reason. So here we would use cochlear implants, as they convert the sound vibrations into electrical impulses, which is what the ear was failing to do on its own, and then the brain can interpret these electrical impulses. And really, that's all nerve impulses and action potential are basically electrical impulses. So that completes this set of slides. It's actually the shortest set of slides in the whole course, so don't get used to just having 33 slides. The next module will be over the cardiovascular system, and I believe there are 85 slides in that, so just get used to having to look at more slides.